Alrighty, so today we're going to talk about DNA replication. Sometimes cells need to make new cells, and when they do that, they need to make sure that they have the equal amounts of DNA in all of the new cells that they make. So they can't just divide in half and shove some of it here and some of it there. They need to have all of the information in both of the new cells. So in order to do that, they need to copy or replicate the DNA that's there. So let's talk about how we do this. So DNA, essentially, when we replicate it, we need to start by opening it up. You need to be able to access that message. So we're going to start by unwinding that double helix. We're going to come in and set little primers on the DNA to tell future enzymes where to go, kind of like little signs or little flags. Hey, come on over here. Then we're going to have an enzyme come in that's going to synthesize a new strand. So they're going to come in and pop on the matching base pairs so that all the base pairs have the correct bonding. And then the thing with that is that it's important to remember is that it's going to look a little bit different on the two different sides. So your leading strand versus your lagging strand, how that happens is going to be a little bit different. So when I said that some people were going to come in and do this and do that, this is who I was talking about. Our major players here, these are enzymes. Yay for enzymes. Now, this might be going back, diving into the archives, but enzymes are made of what kind of macromolecule? That's right, they are proteins. So these proteins or these enzymes are the ones that are going to come in and help DNA replicate. So first, these are in order. Our first friend who comes in, our first enzyme is DNA helicase, and it is DNA helicase's job to unzip the genes, and it will literally come in and unzip those two DNA strands away from each other. It'll separate those hydrogen bonds between the two different base pairs. So DNA helicase is our first friend who comes in. Then you have primase. I mentioned before that you have to set sort of little flags along the strand, the DNA strand that you have opened up. You have to set little flags so that the next enzyme, the next major player who comes in, knows where to go. And that's what primase does, is it lays down those little flags. Then we have our friend polymerase, DNA polymerase. This is the VIP here in that DNA polymerase is the enzyme that comes in and puts in those matching base pairs. So if you have an A, it'll come in and it'll pop in a T. That's right. And then if you have a C, it'll come in and add in a G. That's right. Remember, our curvy letters cuddle. Curvy letters cuddle. Oops. It's not a great looking G, is it? That's okay. All right. And then last but not least, we have our friend ligase. Essentially what ligase does is it comes in and it glues together little fragments of the backbone so that it is all one cohesive piece. So how do they all do it? Well, let's take a look. Now, remember, I had mentioned DNA polymerase is one directional. Our leading strand and our lagging strand are going to look a little bit different on how they get copied. Excuse me, because DNA polymerase can only go one way. Can't go back and forth. So with our leading strand is much more straightforward. With our leading strand, what's going to happen is it's just going to pop the little primer right over here, and then it's just going to read and keep going right along. No changes, no movement, no bumps in the road. Just nice and smooth. It's almost like putting it on cruise control, and it just kind of goes. And that's because it can only read from the 3' to 5'. So this is the direction that it reads. So it's 3' to 5' on the old strand. So it makes from 5 to 3, but it reads from 3 to 5. I think that's the easier way to remember it because the numbers are in order, right? 3 smaller than 5, so it reads 3 to 5. I think is the best way to remember. But on our lagging strand, since it only reads 3 to 5, we don't have that option because our 5' is the one that's there on the end. So it can't just prime it here and then read, right? This is backwards, so it can't do that. So it has to adjust. So what it's going to do is it's going to make it in little chunks. So your primase is going to leave little primers kind of all along so that polymerase can read each little section in between the primers, and then it'll just glue it together when it's all done. So let's take a closer look. So here we have what your DNA strand looks originally. You've got a 5' and a 3' on one end, and then whoop, look, switched. Notice we got the switch on the other side. So what you will have come in first, you'll have DNA helicase will come in and unwind your two sides of your strands. That point at which it is peeled apart, this is called the replication fork. And that replication fork is where it's actually peeled apart. So the helicase will move in that direction this way and keep unwinding. Then you'll have a primer set here. There's our little primer. The primer comes in, tells DNA polymerase, this is DNA polymerase here. It says DNA polymerase, hey, here's where you need to go. And then DNA polymerase will come in and it will read this direction. It will continue to read as helicase continues to unwind. That is over here, this is our leading strand. But our lagging strand over here, this is a little different because here's our exposed 5' end. So what primase is going to do is it's going to come in and lay little primer chunks. So that polymerase will come in at each of these points. So polymerase will come in here, attach, and then read this way. Polymerase will come in here, attach, and then read this way. And what you'll get is something like this. So you'll have these little pieces that get read by polymerase. Each one of these pieces is called an Okazaki fragment because the Okazakis discovered it. Well, it makes sense that you would name it after them, right? When you discover something, you get to name it after yourself. So each one of these is called an Okazaki fragment. And then our last little friend that will come in is called ligase. And what ligase will do is it will come in and just glue these pieces together. Ligase glues them together. It is literally DNA glue. Another important thing to point out is DNA replication is semi-conservative. So what this means is that, you will end up with two strands that have half of the old strand and then half of the new strand. Man, my DNA is not very equal, is it? Try to make this side better. There we go. So you'll end up with two that are equal. They will be much more equal than this. So you'll have one side from the original strand and then one side of the new that was just added on. So this is an important vocabulary word to remember when we talk about DNA replication, that is semi-conservative.